So, as you probably have come to realize by now, it is that a large proportion of the innate immune defense against bacteria and virus is dependent on phagocytosis. And the process of phagocytosis can be divided up in 4 steps. And the first step is the adhesion of the pathogen to the cell which will phagocytose. And this happens, as you saw before with the macrophages, by binding to different phagocytotic receptors, sometimes by complement receptors as a mediator, or sometimes with a Fc receptor, which is a receptor for an antibody, which has bound the pathogen. The second step is the ingestion of the pathogen. The third step is the fusion of the phagosome with the lysosome. The lysosome provides the drop in pH and the pH activated enzymes. And step 4 is the killing and the degradation of the pathogens. Here we also show step 5, which is loading of the peptides onto the MHC class II and antigen presentation, which is not maybe strictly a part of the phagocytosis. So in order to avoid the phagocytosis, the pathogens have developed clever pathways. So how to cope with the phagocytosis? What are the different bacterial strategies? One strategy is to inhibit the chemotaxis, and Neisseria gonorrhea does that. It secretes soluble factors which will block the chemotaxic receptor of neutrophils. The pneumococci can form a capsule around itself, and this capsule is basically a slime ball surrounding the bacteria, making it very slippery. And this capsule formation is very important for the pathogenicity of the pneumococci. E. coli can inhibit adhesion, which is the first step of the phagocytosis. Staphylococcus can inhibit uptake, the second step. Tuberculosis can inhibit the fusion of phagosomes with lysosomes. The lepra bacteria is resistant against lysosomal enzymes and ROS. And Listeria can lyse the phagosomes and escape into the cytoplasm. And many viruses inhibit antigen presentation, which is the last step. So now we have come to the last section of the innate immunity class, and I would argue that this is probably the hardest part. The complement system is something that immunologists all have learned at one point, but they always forget. And we are very bad at this because we don't take it into account when we think about immunology as a whole. And I would like us to try to do that. So the complement system consists of several plasma proteins that they work together to opsonize microbes and to promote the recruitment of the phagocytes to the site of infection, so chemotaxic properties. In some cases, the complement can itself directly kill microbes. The complement works in a sequential proteolysis of proteins, which means that one protein, when activated, cleaves the next protein in a cascade manner. And the products of the complements, they are covalently attached to the microbial surfaces and the membrane, or the antibodies bound to the microbe. And this is, of course, a limitation because the bacteria can just not provide the surfaces which are needed for complement activity, and this is one way to avoid complement. So the complement activation is inhibited by regulatory proteins. So these regulatory proteins are present on the host cells, and this is the basis for the foreign recognition by the complement. So it's again the concept of missing self. The self is inhibiting complement just by having the inhibitor and lack of inhibitor is activating. So the complement is activated through 3 distinct pathways. The classical pathway, it's called so simply because it was discovered first, it uses the plasma protein called C1q to detect antibodies bound to the surface of a microbe or another structure. Once the C1q binds to the Fc portion of the antibody, 2 associated serine proteases called C1r and C1s become active and initiate a proteolytic cascade, which is cleaving complement proteins. The classical pathway, it's the major effector mechanism of the humoral arm of the adaptive immune responses. The alternative pathway, which was discovered later, but it is probably older than the classical pathway since it also exists in the absence of antibodies. Here the complement protein C3 is triggered when it recognizes certain microbial structures such as LPS, and C3 is also constitutively activated in solution at low levels, and that it binds cell surfaces. But when it does, it is inhibited by the regulatory molecules that are present on the mammalian cells, and thus the missing self will cause excessive C3 activation. So microbes obviously don't have this inhibitory proteins, and the spontaneous activation can be amplified on the microbial surfaces. So therefore, this pathway only distinguishes normal self from foreign microbes of presence or absence of an inhibitor. So the third pathway is the lectin pathway, and it is triggered by a plasma protein called MBL, or mannose binding lectin, which is recognizing the terminal mannose residues on microbial glycoproteins and glycolipids. And it's similar to the receptor on phagocytes, which we discussed earlier, which is the dectin-1. The MBL then recruits proteins which are not identical to the C1r and C1s, but similar to the classical pathway, and they will then result in the cleavage of the C3. So all the pathways converge on the cleavage of C3. So once the C3 is sleeved, it results in 2 active components, which is the C3a, which stimulates inflammation and it's soluble, and C3b. And C3b, it initiates the later steps of the complement activation. C3b is then covalently attached to the surface of the microbe. C3b also serves as an opsonine that promotes the phagocytosis and of the microbes, and their receptor recognizing this part of the complement. That is complement receptor 1 or CD35, as it's also called. C3b then binds other complement proteins to form a protease called the C5 convertase that cleaves C5. And once C5 is cleaved, it generates a small peptide called C5a. And the C5a has an analogous function as C3a, which is a pro-inflammatory function. C5b will initiate a complex formation of the C6, 7, 8, and 9, which are assembled into a membrane pore called the membrane attack complex, MAC. And this complex can lyse cells where the complement is activated. So in this picture, we just have a slightly different drawing. Here we start at the C5 convertase and focus on the pore formation. So the C5 convertase is then consisting of C3b, which is covalently bound to the plasma membrane of the microbe, and some other proteins. And then the C5 is cleaved into C5a and to C5b. So once C5b is attached to the convertase, the C6, C7, C8 are recruited. Together with C5b, the C9 will itself be the heart of the pore of the MAC. So on this slide we have a slightly different view of the complement system and the soluble effectors. So in the classical pathway we need an antibody or an antibody complex on a pathogen surface, and C1q will be activated by the C1R and the C1s, and this will in turn activate C4 and C2, and this cascade results in the C3 convertase. So the lectin pathway, the mannose binding lectin, which is called MBL, is not activated by the C1R and the C1s, but from two different but similar proteins, similar function, MASP1 and MASP2, which also then activates C4 and C2, resulting in C3 convertase. And the alternative pathway, where the C3 is constitutively binding to surfaces a bit everywhere, and unless there is an active inhibition of the C3, it will start to form the C3 convertase. And after we have the C3 convertase, we produce C3A and the C3B. And the C3B will in turn activate the C5B, which then recruits the other proteins required for the MAC. And the little A proteins that are the smaller portions of C3A and C5A will function as chemoattractants. They will mediate inflammatory effect and recruit phagocytes and neutrophils. I hope the complement cascades became a little bit more clear. It would be nice to put it more into a context of the other things that we have spoken about before today, and I'll try to do that in the seminar. Right, that was it. Thank you very much for your time, and I hope I've made it worth your while. Also, thank you very much, Mr. Abbas, because the cellular and molecular immunology is a very good resource, and I'm very fond of the pictures in it, as you can see. So if there is no reference to a picture in this lecture, it is most likely coming from the Abbas book, and if not, then I've just forgotten a reference. I will get back to all of you via email and talk to you about the seminar. We will also meet again in the vaccine session. All right.